Hello guys, welcome back to my channel. Today we are going to solve GATE BT 2022 MSQ type of questions. So let's proceed. Which of the following is or are common feature or features for both aerobic and anaerobic bacterial cultures? This is MSQ type of question with 2 marks. First option is glycolysis. Next, NAD plus is the oxidizing agent. Next, oxidative phosphorylation. And last one is 2 net ATP molecules formed per glucose molecule. Respiration process is further divided into 2 type of respiration process. One is anaerobic, that is minus oxygen or in the absence of oxygen. Another one is aerobic. Aerobic means in the presence of oxygen. In anaerobic respiration, glycolysis is found. After that, it goes for fermentation process. This fermentation process is of again 2 types, that is lactic acid fermentation process and ethanol fermentation process. Aerobic respiration shows first glycolysis, after that it goes for Krebs cycle, next it goes for ETC, that is electron transport chain. In between the glycolysis and Krebs cycle, one more step is followed here, that is pyruvate decarboxylation. So in aerobic respiration as well as in anaerobic respiration, glycolysis is common in between these 2. So option A will be right answer. Next is NAD plus is the oxidizing agent. In glycolysis, glucose forms glucose 6-phosphate. After it forms fructose 6-phosphate. After it is converted into fructose 1,6-bisphosphate. Next, it forms glyceraldehyde 3-phosphate and DHAP, that is dihydroxyacetone phosphate. Next, this glyceraldehyde 3-phosphate converted into 1,3-bisphosphoglycerate. Now this forms 3-PJ, that is 3-phosphoglyceric acid or 3-phosphoglycerate. Next, it is formed in 2-PJ, that is 2-phosphoglyceric acid or 2-phosphoglycerate. And last 2-PGA transformed into PAPE, that is phosphoenol pyruvate, and further this PAPE converted into pyruvate, right? This is for the glycolysis cycle. Now, which enzymes or which factors involved in this particular pathway? Here hexokinase is involved and ATP converted into ADP. Here isomerase enzyme is involved. Next, here again one ATP molecule involved and it is formed in ADP and fructose 1,6-bisphosphate, and here phosphofructokinase is involved. Next, in this stage aldolase enzyme is involved which produce glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This is interconvertible, so these are the isomerase. This conversion is possible by the involvement of isomerase enzyme. Now this glyceraldehyde 3-phosphate converted into 1,3-bisphosphoglycerate by the enzyme triose phosphate dehydrogenase, and here one NAD plus oxidized to form NADH plus H plus, and this G3P converted into 1,3-BPGA. This 1,3-BPGA converted into 3-PGA by the involvement of phosphoglycerokinase, and here one ATP is formed by ADP and one phosphate from this molecule. This 3-PGA converted into 2-PGA by phosphoglucomutase enzyme. This 2-PGA converted into PAPE by enolase enzyme by emitting one molecule of water, and this PAPE is converted into pyruvate by pyruvate kinase, and here one ATP is formed. So this is how the glycolysis cycle goes on. This entire event is for one G3P, and if DHAP converts into G3P, this portion will again repeat. So here NAD plus involved to form 1,3-BPGA and side product NADH is formed. So NAD plus is involved in glycolysis. So NAD plus is the oxidizing agent which oxidizes G3P, that is 3-glyceraldehyde phosphate, to form 1,3-BPGA, and it itself reduced. to form NADH plus H plus. So again B option will be right. Oxidative phosphorylation only observed in aerobic respiration. 2 net ATP molecules observed in only anaerobic respiration. So option C and option D is not common in between these two. So option A and B will be the right answer. So this is for today's session. Hope this will be helpful for you. Thank you for watching and keep learning.